There are few ways in which alteration in Hox protein binding or expression might lead to evolutionary changes. These are: first, changes in Hox protein responsive element of downstream genes, changes in Hox gene transcription pattern within a portion of body. Third, change in Hox gene transcription pattern between the regions of the body. And last but not the least, changes in the number of Hox gene. Now this point we have seen that how Hox gene in humans or in mammals are present on 4 chromosomes, how Hox gene is present on only 1 chromosome in Drosophila. We know that in mammals or in man, the Hox gene is present on chromosome 17, chromosome 7, chromosome 12 and chromosome 2, while in Drosophila it is present on chromosome 3 and present on only 1 chromosome. So the change in the number of Hox gene also result in the alteration of the animal's structural formation. Now we will discuss these one by one. Change in the number of Hox gene. If you can see the picture, then broad changes of Hox expression. Can see the segments as well as the appendages. The change in the number of segments, you can see subtle changes within the Hox domain, how there are same number of segment but in one appendages are there and in second appendages are not there. For emergence of appendage, one gene called the distal-less gene DLL gene is responsible for appearance, for regression of wings, that is to suppress the emergence of wing. The ultrapythorex gene is responsible, UBX gene. Now in the fourth picture, the change in the regulation or function of the downstream gene. These are the evident pictures which show that how the Hox gene expression occurs. In the fourth picture, you can see presence of 2 wings in butterfly, that is order Lepidoptera, and presence of only 1 pair of wing and presence of haltere in the third thoracic segment as in dipterans. This is the example of Drosophila. The other one is butterfly. Changes in the Hox protein responsive element of downstream wings. The changes in the genes of Hox protein regulate the ultrabithorex gene expressed in the imaginal disc of the third thoracic segment. In Drosophila, the UBX downregulates certain genes of the imaginal disc, while in butterfly the genes regulated in Drosophila are not regulated in the butterfly. That is, in Drosophila, UBX gene, the product of UBX gene, it downregulates, or we can say suppresses, several genes in the imaginal disc. Therefore, only one pair of wing is present in Drosophila, and third thoracic segment has a vestigial wing that we called halteres, which help in balancing the animal or the bird, balancing the Drosophila. Haltires, halteres. So it helps in balancing the insect, whereas in butterfly, genes that regulate the Drosophila do not regulate in butterfly. So the emergence of two pair of wings, as you can see here, the halteres can be seen like two small hair-like structure. This is the butterfly. Now the second point, change in Hox gene transcription pattern within a region or a portion of the body. Among arthropods, different limb morphology is caused by Hox gene expression differences. Now, what is the meaning of difference in limb morphology? In arthropod, I have already told you that the insects have three pair of legs. Its larval stage may have number of legs, like butterfly has got three pair of legs, two pair of wings, but the larva of butterfly, that is the caterpillar, has several legs. Among the arthropods, differences in limb morphology may be caused by Hox gene expression differences. Insects have 6 legs as adults, with a pair arising from each of the 3 thoracic segments. In Drosophila, the distal less gene, represented as DLL. The distal less gene is important for providing the proximal distal axis of the appendages. The distal less expression occurs in the cephalic, that is the head region, and thoracic region. So we say that the distal less expression occurs in cephalic and thoracic appendage forming disc. But this expression is excluded in the abdomen. We know that the appendages are present only in the head region as well as the thoracic region. There are no appendages in the abdomen. Why? Because the distal less gene is not expressed in the abdominal region. And why it is so? Because in the abdomen there are the abdominal A genes protein expression as well as UBX homeodomain proteins. Thus, the appendages grow into legs and wings in the thorax and into jaws in the head. The distillus gene induces the ventral appendages. Distillus gene expression in is inhibited by the protein products of UBX and abdominal A. And when distillus is not expressed or when it is not repressed, the development occurs differently. That is, when the distillus is not repressed, it results in the development of proximal distal appendages. Meaning of repression is to suppress or not allow to express. So when the distillus is not repressed, यानी जब उसे दबाया नहीं जाता, तो it results in the development of distal appendages. और जब distillus जीन को दबाया जाता है, यानी जब उसके एक्सप्रेशन को इनहिबिट किया जाता है, तो वह डिस्टल अपेंडेजेस के डेवलपमेंट को नहीं कर पाता. So we can say that the distillus expression occurs in the head region and thoracic appendages forming disc, but the expression of distillus gene is excluded, is removed in abdomen by abdominal A and ultrabithorax proteins. In butterfly as well as moth larvae, they are characterized by the rudimentary abdominal leg called proleg. We know that in butterfly, abdomen does not have legs, but as already I have told that the caterpillar or the larva of butterfly, they have rudimentary abdominal legs. Rudimentary means they are not permanent and they will get removed after some time. So they have abdominal legs, as we have seen the legs of caterpillar, how they walk on plants. So this abdominal leg is called proleg. In early portion of butterfly embryogenesis, the DLL expression pattern is the same as it is in Drosophila. During gastrulation, the DLL means distal-less expression is seen first in the head region and in the thoracic region that will give rise to the leg imaginal disc. However, as the development proceeds, the DLL gene of butterfly becomes expressed in third through sixth abdominal segment, third thoracic segment to sixth. There are six abdomen segments, so six abdominal segment. While the DLL expression is seen in both the proximal ring and the sock of true thoracic leg, the expression of DLL in abdomen is restricted to the proximal ring. Thus, in Lepidopteran, that is butterfly, the proleg appear to be homologous to the proximal portion of thoracic leg. इसका मतलब ये है कि बटरफ्लाई में जब डेवलपमेंट होता है और एम्ब्रियोजेनेसिस के दौरान DLL का एक्सप्रेशन हेड रीजन, थोरेसिक रीजन में और लार्वे के थर्ड थोरेसिक सेगमेंट जो आगे चल के बनेगा और सिक्स एब्डोमिनल सेगमेंट्स जो हैं उनमें होता है, लेकिन जो शुरू के ट्रू लेग्स बनने के लिए जो इमेजिनल डिस्क होते हैं वहाँ पर जो एक्सप्रेशन है वह सॉक या SOCKS सॉक्स ऑफ ट्रू थोरेसिक लेग में होता है, जबकि बाकी के जो एब्डोमेन्स हैं उन एब्डोमेन में प्रॉक्सिमल रिंग प्रेजेंट होता है और इस प्रॉक्सिमल रिंग तक ही DLL का एक्सप्रेशन होता है। ट्रू लेग्स जो कि आगे सॉक्स आदि से फिर और कंस्ट्रक्शन होते हैं लेग के, तो ट्रू लेग में सॉक्स ऑफ थोरेसिक लेग पर एक्सप्रेशन होता है, जबकि एब्डोमेन में केवल जो प्रॉक्सिमल रिंग बनते हैं इमेजिनल डिस्क पर से वहाँ पर DLL का एक्सप्रेशन होता है। तो इनके प्रॉक्सिमल रिंग द्वारा जो अपेंडेज बाहर निकलते हैं वह ट्रू लेग्स नहीं होते हैं, वह सिर्फ लेग की भांति प्रतीत होते हैं बटरफ्लाइज में। The presence of larval proleg and DLL expression in butterfly abdominal segment suggests that DLL is regulated differently in dipterans and lepidopterans. ड्रोसोफिला के लार्वे में प्रोलेग्स का निर्माण नहीं होता है। ड्रोसोफिला ऑर्डर डिप्टेरन को बिलोंग करता है और बटरफ्लाई लेपिडोप्टेरन्स को। तो दोनों ही प्रकार के लार्वे को जब हम देखते हैं तो लार्वल प्रोलेग्स मात्र लेपिडोप्टेरन लार्वे में प्रेजेंट होते हैं, जबकि वह डिप्टेरन लार्वे में प्रेजेंट नहीं होते। यह इस बात को क्लियर करता है कि DLL का रेगुलेशन दोनों ही प्रकार के इंसेक्ट ग्रुप में अलग अलग तरीके से होता है। The distilless gene is important for providing the proximal distal axis of the appendages.